Welcome to OsteoBites, and thanks so much for joining us today. My name is Christina Iptoma, and I am mom to Osteo Angel Dillon and Director of Scientific Programs at MIB Agents. And today on OsteoBites, we are joined by Dr. David Ulmert from UCLA to discuss his work on high-throughput characterization of pathobiological responses in osteosarcoma tumors treated with LRRC15 targeted radiotherapy to uncover curative co-treatment approaches. And MIB Agents is proud to support this work with a 2025 OutSmarting Osteosarcoma grant because of Sydney. Thanks so much, Dr. Ulmert, for joining us on OsteoBites today. And thank you also to Andrew Basaga, an osteo warrior and member of our junior advisory board, for being our panelist today. And everyone, please feel free to submit questions during the presentation through the Q&A in Zoom. There should be a little button in your toolbar, and we will share those with Dr. Ulmert after his presentation. A little bit more about our guest today. Dr. David Ulmert is an expert in oncology and biotechnology, specializing in cancer biomarkers and targeted therapies. He developed high-affinity antibodies against androgen receptor-regulated enzymes HK2 and PSA, now in clinical trials across the U.S., Europe, and Australia in collaboration with Janssen for radioimmunotheranostics, CAR-T therapy, and bispecific targeting. His lab also developed DEMP19, an LRRC15 targeted antibody licensed to Lantheus, with a phase 1 trial in osteosarcoma that just recently opened. And Dr. Ulmert leads UCLA's preclinical theranostics program and conducts population-based studies on cancer biomarkers and risk factors with international collaborators. And he's widely recognized for advancing prostate cancer research and translational immunotheranostics. And we're so glad he's now working on osteosarcoma. We're thrilled to have you on OsteoBites today, Dr. Ulmert. And Andrew, could you go ahead and introduce yourself?
Yeah, hi, my name is Andrew Basaga. I'm from the Chicagoland area. In 2017, I was diagnosed with osteosarcoma in my proximal femur. I finished up the standard MAT chemotherapy treatment in 2018 and have been NED ever since. So I'm going on about 8 years of no disease. Like Christina said, I'm a member of this past year's junior advisory board. And I'm also a student at Creighton University studying biology, and then hoping to go to medical school after. So with that, I'll hand it off to Dr. Ulmert.
Yeah, thank you so much for inviting me to share some of the work that we have been pursuing here over the years. Before I start, I would like to, thank the Kaplan family for their funding that has been supporting some of this work that I'm presenting here today because of Sydney. And I also would like to underline that the work that I'm presenting is a result of teamwork, not only together with colleagues at UCLA, but massive support and input from other universities and other groups, such as Lund University, University of Essen, WashU in St. Louis, MD Anderson, University of Miami, Sloan Kettering, and in Finland, University of Turku. So for those of you who are not familiar with the concept of radioimmunotheranostics, it's when you utilize an antibody or a ligand that targets a specific molecular molecule on the cancer cells. And then you're utilizing that vehicle, that ligand and the antibody as a vehicle, and you label it with either a diagnostic or a therapeutic radionuclide, so that the vehicle is then trafficking and taking the diagnostic or therapeutic radionuclide specifically to the tumor cell that is expressing the molecular target that is of interest. That offers us a unique opportunities to not only diagnose cancer that is expressing the molecular target, so you can see with a PET scan or a SPECT scan where the molecular target is located, meaning where the tumor is located. And based on those images, you can select patients that would be eligible for therapy using the same vehicle, but you are then exchanging the diagnostic molecule to therapeutic radionuclide, meaning that the therapeutic radionuclide is toxic, so it is irradiating the cells specifically where the molecular target is located. So instead of sort of treating the whole body or surrounding tissues as well, you're treating only where the molecular target is located. A very good example of this is PSMA-targeted radioimmunotherapy. or targeted radioimmunotheranostics, where a small peptide or a antibody is used as a vehicle for targeting PSMA. And here's an example when you're utilizing that vehicle targeting PSMA for diagnostics. As you can see here to the left, you can see that the tumors are lighting up as black spots, and you can see that where the black spots are located is where the PSMA is located. In this case, the patient has very generalized disease with lots of metastases in the skeleton, and then you take the same vehicle and you apply a therapeutic radionuclide, give it 2 cycles of this, so you're irradiating where the PSMA is located. And as you can see, when you do the next imaging of the patient with the diagnostic radionuclide, you can see that you have significantly decreased the tumor mass and the tumors that are then expressing PSMA. Continuing with the therapy for another 2 rounds and doing another round of imaging, you can then see that the tumor compared to the first image has really significantly decreased uptake in the tumors, and the tumors are then treated. So the target that we have been going for is, instead of PSMA, as I had an example, our target is LRRC15, and we're targeting with an antibody. So a little bit about LRRC15. It's cells that are either cancer cells that are originating from mesenchymal stem cells, or it's mesenchymal stem cells that has been migrating to tumor tissues. So mesenchymal stem cells are normally found, or mostly found, in bone marrow, and they can either divide and become tissues such as bone, muscle, cartilage, but they're also central in healing tissues. So if you have a wound, the mesenchymal stem cells are then migrating to the wound is located, and it's helping the wound to heal. So if you look at the tumor as a never healing wound, when the mesenchymal stem cells are then migrating to the cancer cells in the tumor, the cancer cells are then expressing molecules and other things in the environment that sort of hijack the mesenchymal stem cells. And the mesenchymal stem cells then starts to combat the tumor, but it's also starting to help the tumor to form supportive tissue that is then making the cancer more aggressive over time. So LRRC15 is expressed either on mesenchymal stem cells that are malignantly derived and directly, such as osteosarcoma, but it's also mostly expressed in the supportive stromal tissue in very aggressive cancers when they have metastasized often and are treatment resistant, such as breast cancer, gastrointestinal cancer, pancreatic cancer, etc. So it's either found in the stroma directly, or it's found on the cancer cells, and or both, such as in osteosarcoma. So it's driven by TGFβ, and TGFβ is sort of a master molecule that is doing a lot of things in the body. It's controlling cell behavior, tissue repair, and immune system responses. Originally, when the tumors are new or they're less malign, TGFβ is actually helping to trying to eliminate the tumor. But TGFβ at some stage goes from being a friend to foe. And in those cases, when sort of TGFβ switches to the dark side, that's when we start to see LRRC15 expressed in the tumors. So here's an example of TGFβ expression in normal tissues. As you can see, there's no expression in brain, liver, lung, and colon. LRRC15 here is brown. And you can see a little bit of LRRC15, but very little in wound tissues and some in hair follicles. But apart from that, we don't see LRRC15 expressed apart from in tumors. As I said before, the most common is that LRC15 is expressed in stromal tissues, in the stromal compartment of tumors. And here are some examples. You can see that the cancer cells are not expressing it, but it's the stroma around it, which is supporting the cancer cells that are expressing LRC15. In then mesenchymal stem cell derived cancer cells, such as osteosarcoma, glioblastoma, melanoma, LRC15 is not only expressed in the stroma, but it's also directly expressed in the cancer cells. So in the case of LRC15 and osteosarcoma, we're not only targeting the cancer cells, we're also targeting the supportive stroma. But LRC15, why LRC15 is so interesting is based on some fantastic work done by Shannon Turley's team at Genentech. She found that LRC15, together with 10 other genes, are the main driver for resistance against immunotherapy, such as immune checkpoint inhibitors. And she has shown, and we see that in the picture where it says in the B picture, you can see that LRC15 here is violet, and you can see that in the stroma surrounding the cancer cells, we have LRC15, and it's producing sort of a barrier for the immune cells to interact and try to combat the cancer cells, but it's also producing lots of other factors that is inhibiting the immune system from attacking the cancer cells, and it's also doing other things such as making it more aggressive and metastasize faster. As we can see to the graph to the left, that the higher TGFβ expression you have, the more LRC15 expression you have in the tumors. So they made a very artificial model, tumor model, where they could turn on and off LRC15 in tumors. And when they turn on LRC15, you see that in the black curve, you can see that that's when TGFβ is turned on, and in the blue, that's when TGFβ, they're turned off TGFβ, and then they treat with checkpoint inhibitors, and the checkpoint inhibitors becomes much more efficient when treating tumors. So we started to look into osteosarcoma because we thought it was very obvious, because it's expressed both in cancer cells and in the stromal tissue. And what we did is that we went to a biobank here at UCLA, and we looked at primary biopsies, so when the patient was diagnosed, and looked at LRC15 expression, so we stained for LRC15. And then we compared to survival in these patients. And what we found is that patients with very high or high expression of LRC15, already at the time of diagnosis, they are unfortunately less sensitive to chemotherapy and therapies overall, and they are succumbing much faster compared to patients with low or no LRC15. You can see that Kaplan-Meier curve in overall survival, how it's dropping very rapidly in patients with high LRC15 expression. So we are utilizing and developing an antibody that targets LRC15, and we want to use this as a vehicle for radioimmunotheranostics. And we think that radioimmunotheranostics is especially important when it comes to LRC15 because it's not expressed in all tumors and it's heterogeneously expressed as well. So what we did is that we want to confirm initially that the antibody is not only specifically targeting LRC15 on the cells, but also that the number of LRC15 molecules on the cells is then correlating with the ability for the antibody to target and bind to the cells. And that we see here, we compare then the genetic expression with RT-PCR with the number of LRC15 molecules per cell that the antibody is binding to. And to the right, you see confocal microscopy at room temperature, incubated with antibodies incubated at room temperature. DUNP19, which is the LRC15 antibody, is green here, and you can see that it's outlining the cell. What's unique with DUNP19, it's also that does not only have very high affinity, a very high specificity for LRC15, but it's also that when it's targeting LRC15, it's internalized. And that is important because since we're utilizing the antibody as a vehicle for radionuclides, we want the radionuclides to get as close, especially when it's a therapeutic radionuclide, as close as possible to the DNA, because the radionuclides are emitting either alpha or beta particles, as the beta particles we've mostly used, and they are causing DNA breaks. So you want to be as close as possible to the DNA. And the other thing also that you have a higher retention, so the antibody and the radionuclide is staying longer in the tumor if the antibody is internalized by the cells. So you can see here that clockwise, when you start at 4 degrees and you incubate the cell with the antibody, which is 4 labeled, that when you increase the temperature and the cell sort of starting to be active again, that the antibody, which is in yellow, is then taken up by the cell and it goes inside of the cell. So we started by labeling the antibody with a positron emitter, and the positron emitting radionuclide is something that you can utilize, that is utilized because it is emitting positrons, and then you can utilize for something that's called PET imaging, meaning that you can use this PET machine or PET camera to see exactly where the antibody and the radionuclide is located in the body. And that gives you an ability to diagnose, in our case, and see where LRC15 is expressed. So this is one of our mouse models that we started with, is an osteosarcoma tumor called SAOS2. It has sort of a medium expression of LRC15. We have then inoculated these cancer cells on the flank of the mouse. It forms a subcutaneous tumor. And then we injected in the tail vein the antibody that's radiolabeled copper-64, which is the positron emitter. And over time, you can see the sort of the color blob on the mouse here, how at 12 hours it's taken up and more over time, but it's then decreasing in the rest of the body. And to the left here, you can see when we compare it to a sodium fluoride scan, which is only showing sort of bone density, because these are very bone dense tissues, that our antibody is performing better with what is used also in the clinic. So when we apply then a lutetium-177, which emits both photons and beta particles, the photons can be used for SPECT imaging, with just another type of camera where you can see where the radionuclide is located. And it has a beta particle emitting capacity, meaning that it can also irradiate and kill the cells by DNA breaks on the DNA. So this is a complicated graph, and it's how we are evaluating our targeting capacity. So what we do is that we take a group of mice, we are injecting them at the same time with the radiolabeled antibody. And then at different time points, we're selecting groups that are being euthanized, and we're then dissecting the mice and we're measuring the amount of radioactivity in the different organs at that time point, and then next time point, and then next time point, and next time point. These mice are carrying tumors, so what you can see here is that it decreases over time in organs and that are not expressing LRC15. So it decreases, in all other types of organs, but in the subcutaneous tumors, it is then staying in the subcutaneous tumor. Here's U118, which is a glioblastoma cell tumor, and here's SOS2, which is sarcoma. And here's also then H09, which is also an osteosarcoma cell line. And these are expressing different amounts of LRC15. And we can see that the higher LRC15 levels that they are expressing, the higher is also the uptake of the radionuclide and the antibody. Down here, we did tons of tests to show that it's specific. We used a non-specific antibody, which we compared to the specific antibody. And we also then further tested at 72-hour time point that the uptake is correlating with the expressed amount of LRC15. So when we have the PET camera and we're imaging, we can not only localize where the tumor is, but we can also quantify the amount of LRC15 that is expressed. And since that is correlating with aggressiveness, it could be further a parameter for diagnosis and how to monitor patients during therapy. When we then look at how well it's treating osteosarcoma, we started by looking at utilizing a tumor based on HUO9 cells that inoculated subcutaneously. We gave a dose of the antibody in the tail vein, the radiolabeled antibody, when the tumors are sort of medium-sized. And we see then here in the blue line that it is hindering the tumor from further progression. And here, when the tumors are really big and we're giving a dose, we see that it is better than the black graph, which is when it's untreated, that you are decreasing the progression over time. But it's still not treating as well as it does if the tumor is smaller. So then we wanted to, of course, evaluate that if we not just give one dose, but we give multiple doses over time, how well the therapy is performing. In that case, we want to do that because we want to see if it's tolerable for the mouse or the model. But we also want to see that if we see early signs of resistance towards the therapy. And as you can see here, when we start the treatment, that you have good effect, and when you inject the next one is tolerable, but the tumor is actually keeping, not progressing, but it's not sort of completely going down to zero either. And the black graph here is when we're not treating the tumor at all. And then we have the Kaplan-Meier, the survival curve, so the percent survival here on the right axis. You can see that the untreated in the black, they are succumbing fast. But the treated tumors are staying alive and surviving over the time that we observed mice. So then we're, of course, interested, the tumor that was left, what's that? What does it look like? So we took tumors that had not received any treatment before. We injected it with the radiolabeled antibody. We took out the tumor and looked at with a SPECT camera so you can see the radioactivity. And then we also did a CT. And the CT is good in this case because you can see the density of the tumor, and that is reflecting how calcified the tumor is. And since it's an osteosarcoma tumor, we have a bit of calcification already at the start. But when you give it then 3 treatments, what we see is that mostly what's left and why it's not moving further down to zero is that bone is, instead of forming normal scar tissue, it's forming bone and it's calcification tissue. So bony tissue that is mostly innate. And as you can see, there's still a little bit left of LRC15 taken up, but it's very minimal. And that tissue we also have analyzed further that I'm going to present in a couple of slides. So then we did an orthotopic model instead, because of course we wanted to see what does it look like if we inoculate the cells into the bone tissue instead, into the tibia. So here is an example where we inoculated the cells into the tibia. The tumor here that the red arrow is pointing at is a very small tumor, but still when we inject the antibody that is radiolabeled, you can see here that regardless that it's a tiny tumor, we can very clearly detect the tumor. And also you can see that there's nothing else in the body. It's strictly going to where the tumor is located. Then we took this model and we see, how well can we treat these tumors that are more resembling sort of primary diagnosis or primary location or bone metastases. So we know that these cells are really aggressive and 100% usually of the mice that we inoculate, they are forming tumor tissues. So we inoculate them, we waited for 21 days, and then we gave all the mice, or half of the mice, a dose of the radiolabeled antibody. And then we did a SPECT imaging as well, because as I said before, you can utilize lutetium-177 both for SPECT imaging and for therapy. So it's doing both. And that gives us an ability to see the tumor right after we have injected it. And then we did a follow-up scan 163 days later to see what has happened to the tumor and if we saw any uptake. And what we saw at the start, you can see the uptake of the radiolabeled antibody located in the tibia here. Here it's an example of 2 of the mice. And then we did a new injection then after 163 days. And what we can see is that we don't have any uptake of the antibody anymore. And it looks like the the tumor is completely gone. Here is the survival curve. The green is the mice that received treatment. You can see that one of the mice died, actually not due to any reasons that has to do with the tumor, was something else, which also happens with mice because these are immunosuppressed mice, and accidents happen or they die because of other reasons as well. But they all survived during the time that we monitored them for almost 200 days. And while the other untreated tumors, they succumbed over time. We then wanted to evaluate that it's not only osteosarcoma that we can targeting, we can also target other LRRC15 expressing tumor cells. So here's U118 subcutaneous tumors, so flank tumors, and it is expressing LRRC15 both in the cancer cells and the stroma, not as high as the H09 cells and the H09 tumors, but it has high and targetable LRRC15. As you can see here, because it's different biology as well, compared to bone tissue or bone tumors, that when we give the first dose, it goes to 0, and they get another dose as well. But it starts to sort of go up again, and it's a small tumor that is not progressing, but it's still there. Again, something we're very interested in seeing what's left and what it'll look like and what's the genetic profile of those tumors. But you can see the right, so the black graph here, when we look at the tumor progression over time and tumor volume over time, that they are increasing and succumbing due to the tumor mass. We also did it in a model which is called HCC 1954, which is a breast cancer model. Those tumors have very low expression of LRRC15, but only in the stroma, not in the cancer cells. And here we gave 1 dose and we studied the effect. And you can see that even though the LRRC15 expression is low, we still have a quite good tumor response when we're treating it. So going back to this, we want to see in these tumors that we're treated, what has happened, what does it look like, and especially what has happened to, as I mentioned before, that Shannon Turley's team has shown that LRRC15 is part of a genetic profile that has been a set of genes that has been correlating to immunoresistance and immune checkpoint resistance. So we want to specifically see, when we're targeting the LRRC15 expressing tumors, are we also ablating and eliminating the LRRC15 and the other genes that are associated with immunotherapy resistance? So we did bulk RNA sequencing. And what's great with mouse tumors in this case that we're inoculating human cancer cells in a mouse is that the stroma that forms the stroma in the tumors, that's murine tissue. And while the cancer cells are human cells, so we can separate the two, so we can separate what we see in the stroma and what we see in the cancer cells. So that's pretty neat. What we see here, this very complicated graph that I'm trying to simplify, but what we see in U118 and LRS and HU9 is that we see a downregulation of LRRC15. when we're giving the treatment. And in HCC 1954, because it's cancer cells, we don't see anything because they didn't express any LRRC15 in the cancer cells. We can also see that the TGF-beta, because TGF-beta can sometimes, when you give irradiation, it's responding by increasing TGF-beta as a response to radiation. So we want to see that TGF-beta is not increased over time, which could potentially then further boost sort of the LRRC15 expression, given that LRRC15 is driven and regulated by TGF-beta. But what we see is that TGF-beta is going down in or decreasing in HU9 and U118 models, but we see no significant difference in the HCC 1954, where it's only expressed in the stroma. When we're looking specifically at these 11 genes that Shannon Turley's team looked at as a correlation to resistance, we're also seeing that overall in the cancer cells, when we separate the cancer cells and the murine tumor stroma cells, that we're ablating almost all of the expression of these genes in the tissue that is left after we have given the therapy with radio-labeled antibody targeting LRRC15. And we see the same thing in murine tumors. So red is high expression, blue is low expression in the cancer cells, and then red or orange and is low in murine tumors, but green is high expression of LRRC15. So we're decreasing it in overall in almost all of the genes that she said is correlating, where she has shown is correlating with immune checkpoint resistance. So then, of course, there comes the big question: well, if you're downregulating the LRRC15 and the other genetic other genes that are correlating with immune checkpoint resistance, will you have a better effect with immune checkpoint resistance? As she had shown, like the Turley team has shown, when they are sort of upregulating and downregulating the LRRC15, but doing it in a different way. What we see here is where we take a very aggressive syngeneic model, which means that it's murine cancer cells that you put and inoculate into a mouse with a full immune system, because you need that if you want to test the immunotherapy compounds that we utilized. And here is then a mouse in blue line, so you can see that it's rapidly increasing in tumor volume over time. And here is then when you give a combination of not only one ICI, immune checkpoint inhibitor, but actually both. And we can see that, you have a significant shift, that you're decreasing the tumor progression. And then red is when we give one actually very low dose of LRRC15 targeting radioimmunotherapy. But the big sort of revealing graph is the one to the right here, which is when we are combining the two. So we see that the downregulation of the ICI resistance gene profile that we're downregulating with the therapy when we're targeting it with an antibody that's radio-labeled and give radiation to LRRC15 expressing cells, that all of a sudden you get a much better response to immune checkpoint inhibitors. As you can see, that we gave them one dose of LRRC15 radiotherapy, and then we're combining with 3 rounds of checkpoint inhibitors. And what we see is that almost half of the mice that we studied, they were completely cured, and we saw no tumor at all when we combined the two drugs. So actually, the 3 drugs here. We then tested in another syngeneic cell line, which is even more aggressive, and has actually even lower LRC15 expression and again, just in the stroma, not in the cancer cells. We see that we're not treating with anything, just water, we see nothing is really happening. When you're treating with the checkpoint inhibitors, you see that you have an effect. You have a similar effect with when you treat with a low dose of the radiolabeled LRC15 antibody. But when you're combining the two, you see a much better effect, and you clearly see that the immune checkpoint inhibitors are becoming much more effective when you are co-treating them with the LRC15 radioimmunotherapy. So one of the major aims that the Kaplan family has generously sponsored us with because of Sydney, we wanted to further investigate how TGF-beta is regulating LRC15. That is important because it gives us an ability to better understand the pathobiology of LRC15. In previous studies that had looked at TGF-beta so the TGF-beta regulated expression of LRC15 has been in mesenchymal stem cells that they have incubated with really high levels of TGF-beta over a very long time, which is not correlating to what we have in the body at all. And they see slight increase of LRC15 when they're doing that for a prolonged time. So instead of looking at stem cells, we want to look at the cancer cells. What we found when we evaluated a number of cell lines that are expressing LRC15 in different amounts, we starved them from TGF-beta first, meaning that you have a low LRC15 expression. And then we gave TGF-beta back. So we measured LRC15 under the starved condition where there's no TGF-beta. And then when we give TGF-beta back, and we thought that what we're going to see is that they were all going to increase in LRC15. But we didn't see that. We saw that, half of the tumors, they increased really rapidly in LRC15 after giving TGF-beta back. But some of the cells, we show basically no change at all. And that was very interesting to us because when we give a drug that is inhibiting TGF-beta, the TGF-beta receptor, we see that they all go down in LRC15 expression. So the upregulation, meaning which is the interesting part because LRC15 is correlated with more aggressive disease, more resistance and immunotherapy resistance. So we wanted to see that this sensitivity and the profile of these tumors to be more sensitive to TGF-beta and produce even more LRC15. What is the cause of that? That can help us when we are evaluating PET scans, for example, with patients. If you're monitoring the patient over time and you see that LRC15 is increasing, you want to know why it's increasing. And also, apart from targeting LRC15 with the radioimmunotherapy, are there other drugs that we can utilize to inhibit TGF-beta from upregulating LRC15? So with this, in a number of steps, we did something called functional compound screening, which sounds complicated, and it is. But what we did is that we took the cells that we have starved that had an upregulation of LRC15 under TGF-beta. We starved them from TGF-beta, and then we treated them with a library of drugs, 1,800 compounds. And then we wanted to figure out, because these compounds here have been very well characterized, so we know what they're doing and which pathways they are sort of attacking. We wanted to see which of these compounds are then hindering TGF-beta from upregulating LRC15. We also did single-cell RNA sequencing where we compared the non-responsive TGF-beta to the responsive TGF-beta LRC15 cells. So the cells that when we starved them from TGF-beta, we gave TGF-beta back. and we saw no sort of change in LRC15 compared to those when we starve them, give TGF-beta back, and we see an rapid increase of LRC15. We're going to compare and see what's the difference between these groups of cells. And then we want to then combine the information computationally to see what information from these two modalities of evaluating LRC15 regulation, what can we get out of that? And that's what computationally done through our amazing collaborators in the Graeber lab and in the Yang lab. So don't ask me many questions about computational biology, because I'm not the expert. I just know that it's some type of magic that they do in the computer, and you get a good result out of it that is often interesting. And then, of course, when we've singled out the genes that were sort of potential regulators of LRC15, we wanted to see that it's not just a result of computational biology, but is actually having an impact. So what we did is that we starved, again, the cells from TGF-beta, which treated them with siRNA knockdown compounds, meaning that we're hindering the gene from expressing the protein, or the protein that we think is part of the regulation of LRC15, and seeing if it has an impact of hindering TGF-beta from upregulating LRC15. So here is an identification of, or is a sort of a cartoon of our strategy. And this looks simple, but it took about 1 to 2 years to establish this assay. So that was done by Claire Story together with Robert Dominiceau at UCLA. And they then, again, starve the cells, they add the compound, then they add TGF-beta, and then they are then staining for LRC15 on the cells, and then they're using a confocal microscope to quantify how much LRC15 is then do you see when you're treating with the drugs. So what you can see here is like, this is before, and we've starved the cells from TGF-beta, and we are then staining for LRC15 here in the middle, that's purple. And then when we're adding TGF-beta back, you can see that you have a much higher expression of LRC15, and then you quantify and compare these. And then, of course, in the middle of when you're evaluating the compounds, you're adding the compound in between, and then evaluating and quantifying LRC15 expression. So out of the 1,280 compounds that we tested, 282 of those were non-toxic. The other were when we applied it to cells that were just toxic to the cells, so it didn't give us any information. So we further looked into these 282 non-toxic compounds, and then we found 72 of these were hits, meaning that they inhibited TGF-beta from upregulating LRC15. And what we found is that the drugs that were doing this were a wide range of types of drugs, but they were they were immunomodulators and different cytokines, etc. And then that also shows that how wide TGF-beta's function is, and how it can be affected by many types of compounds. But it still gives us interesting information that we can then utilize together with computational biology with our single-cell RNA sequencing. So here is then when we compare single-cell RNA sequencing, when we're doing comparing the TGF-beta inducible versus the non-inducible. What we see here is that the inducible, they have an upregulation of genes related to cell cycle and immune signaling, which was a relief, because then it's what sort of Shannon Turley has shown before, and what we have seen in our animal experiments, meaning that LRC15, higher LRC15 and sensitivity to TGF-beta is associated with immune signaling, immune response. We also see the same in innate immune response and apoptosis to to some degree as well. Let's see. And then we did, as I said before, we add for our sort of potential regulators, we took an siRNA construct to knock down the protein before we gave TGFβ back to see if the single potential genes that are regulating had an effect on upregulation of LRC15 when we stained. Let's see. And what we found is that in the 3 cell lines that we tested, that one of them, EID1, when we knocked down that one, actually it increased TGF-beta, so it did the opposite. 3 or 4 of the genes that we singled out, they did not alter the TGF-beta. But what we found was that MMP, Spark, TGF-beta receptor 2, and Wnt5b, they significantly reduced LRC15 or reduced TGF-beta's ability to upregulating LRC15. The TGF-beta receptor 2, it's kind of logical because it is the receptor for TGF-beta, so it amplifies and upregulates LRC15. Given that it has more receptors, there's more for TGF-beta to interact with. The others were very interesting, MMP2, Spark, and Wnt5b, because they are promoters for tumor aggressiveness and immune evasion. So basically what's required for, if we make a very short summary and assumption, is that in order for TGF-beta to rapidly upregulate LRC15 and making the tumor more aggressive, you already need to have certain expression of genes that are related to tumor aggressiveness and immune evasion already there, which shows, again, that it's really, LRC15 is really a biomarker from when TGF-beta goes from a friend to foe and starts promoting tumor aggressiveness and immune suppression. So in summary, we have then made we've constructed a precision targeting and tumor and supportive tissue that are expressing LRC15, which in the case of osteosarcoma is expressed on both the stromal cells and on the cancer cells. So hopefully all of the tumors is attacked and can be irradiated with specifically non-invasively with this radiolabeled antibody. And as a biomarker, what we found is that TGF-beta, we knew that LRC15 is induced by TGF-beta, and that high LRC15 levels reflect TGF-beta activity, but it also depends on other promoters of aggressiveness and immune evasion that needs to be there before TGF-beta can really rapidly upregulate LRC15. And of course, this is what we saw here. It's probably many more factors that are involved in this. But from our studies, we can assure that these are involved in the regulation. What we've also shown is that when we take these 4 genes plus LRC15, and when we correlate it with Shannon Turley's 11 gene panel, that they are actually perfectly correlated, which further prove that it is related to immunosuppression and resistance to immunotherapy. So when we are applying LRC15 and we're attacking LRC15 and ablating LRC15, what that leads to is that, yes, we are decreasing the tumor mass, but we're also able to detect and dismantle the protective barrier that is hindering the immune system's ability to recognize and destroy tumor cells. On top of LRC15 also being a marker of aggressiveness and resistant towards other therapies when it comes to osteosarcoma and when it comes to other types of cancers. And what's great here is that thanks to Lantheus, through their really hard work led by Asim and Anand, we have now gone from taking this project from the bench to the bedside. Again, an enormous effort from their side and a lot of paperwork. And of course, together with Noah Fetterman, who's going to lead this study at UCLA, we hope that it will be a successful trial and that we will be able to see a change in the progression and the ability to treat these patients. It is a study that is going to be at UCLA, again, collaboration between the nuclear medicine department here at UCLA and Noah Fetterman. We are going to start, and we with Lantheus and Noah Fetterman are going to start by looking into patients that are older than 18 and test dose escalation and test toxicity and the ability to target LRC15. And we're validating that based on, again, the SPECT images so that we see an uptake in where we have previously found tumors through other anatomical modalities such as CT and MR, etc. We're going to start with 30 patients in the first cohort, and it's 6 sites that are helping to recruit patients. When this has been confirmed and done, we are going to go to dose expansion and include another 4 sites and another 25 patients. Again, the objectives of safety, tolerability, efficacy, pharmacokinetics, biodistribution, dosimetry, and pharmacodynamics. And there are, of course, more information about this trial. And if you want further information about this trial, you can either contact Carrie Sforzo at Lantheus, or you can reach out to Noah Fetterman. And I'm sure that Christina can also help you to get in contact with the people who are running this trial and so forth. Yes, so that's what I had to present here today, and I'm glad to take questions if you have any.
Thanks so much for that amazing overview. What a great explanation, and thanks. You just kind of made that very easy to digest. So thank you for that. And also so exciting because often we'll have, great study presented with really with data that looks really great. And so it's so nice that there's actually a clinical trial, because often the question is like, okay, that looks great, but when can we get this to patients? And so yeah. So nice that it's already, there's a trial available. So we actually got a question already in. If Andrew, you want to take that one.
All right. One of the questions. So Dr. Ulmert, what are the advantages of your approaches compared to an ADC?
Well, an ADC has been already been tested against LRC15, and they didn't show any efficacy. I think there's probably room for ADCs as well, but ADCs are very particular in going for a specific inhibition. With radiation, you're able to actually select the patients that have LRC15 expression. It's still very heterogeneous, and you don't see expression of LRC15 in all osteosarcoma patients. But it gives us an ability to select the patients by imaging and then applying radiation, which is more broadly causing DNA repair effects or DNA destruction. And then you can follow up with further imaging to monitor the treatment efficacy. I'm sure that there are room for ADCs as well, but I think that it's the benefit of being able to select the patients is better than an ADC, yeah. So I think that what is going to be super interesting here is that we're going to biopsy these patients before and after LRC15 radioimmunotheranostics. And we're going to evaluate what happens to the tumor. And it could be that you have some sort of change that in the tumor that can be either maybe further treated with another type of drug or a specific ADC to target LRC15. So there's probably room for that too. But again, I think that the selection of patients and the specific irradiation is more effective and easier to evaluate at this stage. So yeah, I think that this is that's the overall sort of point. We're also talking about what happens to the tumor. We are now in the second aim of my osteosarcoma Hero Award funding is to study what happens to mice, because we only studied syngeneic mice of other tumor models that was not osteosarcoma. But now we've set up a collaboration, so we're going to test what happens to syngeneic models if we see the same effect as we saw with the other cancer models, how it changes the tumor microenvironment in the tumor, and that is going to give us the ability to give other co-therapies and also correlate with what we will see in the clinic. And maybe there's like a perfect toxin that can be put to the LRC15 antibody as well to co-treat. But I think that the easiest and the more effective in this case is to utilize radiation.
Can I ask a follow-up question about the imaging? So you're using imaging, first of all, to just see how many cells express LRC15. How do you translate that to a number? Is it similar to staining? Is it can you compare that apples to apples? And also, would imaging be better at kind of capturing that heterogeneity versus a tissue sample, since you're not limited to just that tissue sample?
Yeah, I mean, you can do both. So first of all, it's non-invasive. So when you can see how many of the tumors, where they're located, and the individual LRC15 expression in the individual tumors. And you can combine that with a targeted biopsy to confirm it as well, because you know where the tumor is located. But yeah, it gives you an overall image. Of course, utilizing lutetium SPECT is awesome that we're doing that, and that's fantastic. And it's easy that you have just one type of radionuclide that you apply for both imaging and therapy. But you can also exchange it, as we've shown here, to zirconium for PET imaging and or copper, and which are more easy to quantify and gives us slightly better images compared to lutetium.
Oh, okay. Gotcha. And let's see. Oh, there's another question that came in. Andrew, do you want to take that one?
Yeah, sure. So Dr. Ulmert, are there any other emerging trials regarding this target or other related pharmacological targets? And is Shannon Turley pursuing this clinically, either at Roche or now at Amgen?
Not to my knowledge, she's not approaching this. But again, Genentech is a pharmaceutical company, and they keep their strategies very close to themselves. I had a conversation with them, and I shared all of my data and all of my findings, but I didn't really get much back what their strategy was. But yeah, I mean, they've had a long time because they've known this relationship for a long time, that LRC15 is correlated with immune resistance. But yeah, I saw that there is another type. I don't know exactly what it is, but it's another study, I think in China, that is opening for another LRC15 antibody type of approach for LRC15. And again, we were not first to target LRC15. There was this ADC compound. The difference between our antibody and their antibody is that their antibody is not internalizing LRC15. They were not selecting the patients based on LRC15 expression. So you're treating patients that potentially have no effect, and include that in the trial. And also, I think that now ADCs have become a little bit better with linkers, but you also have a problem with ADCs that they are more vulnerable to degradation in the blood circulation than with what we have with targeted radioimmunotheranostics and utilizing radionuclides. But again, I think that internalization is key, especially for an ADC, that you are delivering the toxin inside of the cell instead of delivering it in the sort of the outside the cell and in the tumor microenvironment. So in that case, I think that our antibody is superior to what they are using. And I think that their trial, I'm just guessing, and you know, there's so many, I think it's AbbVie, and there's so many talented people at AbbVie, so they probably have a rationale for it. But I think that selection of patients is key. We've seen that with PSMA, for example, where if you don't have an uptake of PSMA on the PET, you're not eligible for treatment, instead of treating all prostate cancer patients with metastases. And again, you can follow up with an imaging component, so you can see if you see any, if it's effective or not, by, as we shown with the images with the PSMA, and monitor the treatment. But no, I don't know what Genentech's approach and what they're going to do. If anyone knows, you know, I'm very interested to hearing what their approach and strategy is. But I have to thank them. You know, they have produced fantastic articles on LRC15, two back-to-back Nature articles and one cover story in Cancer Discovery, papers that I wish that I would be able to write myself.
Well, you wrote a great paper too, which you and your team, which we shared a link in the chat too. Dr. Ulmert, do you mind if we keep you for a few more minutes? I know we're a bit over time, but I can give a few more questions.
No, sure. Go ahead.
Great. Andrew, take it away.
Alrighty, next one. Do you think the payload made a difference with the ADC?
The payload, again, ADC is not, I think that it's better if you ask someone who is an expert on ADCs. I can't really comment on of course I think that if you pair up imaging, like this is very academic and theoretical, but if you pair up the imaging portion, and you are doing targeted biopsies of the different tumors that are expressing LRC15, and you are then genetically looking into and profiling these tumors, you can probably tailor the ADC and the toxin and the payload that you're giving more effectively. Again, we are not, so we don't have that problem because we give radiation and it's very simple. You are sort of destroying the DNA and that's it. Either you are using we all also have different types of payloads that we can use, a different type of radionuclides. So we both have betas, which are not as if, they're effective, but they have a very long range. so that if you target, which is called bystander effect, that you're targeting one tumor and the radionuclide is going to that tumor cell, but given the range of the betas, you're also then irradiating cells around you that have a lower LRC15 or no LRC15 expression. So if you target the stroma, for example, you're destructing the stroma, and that might, of course, lead to that the cancer cells, because they're depending on the stroma, that they are not surviving because of that. But it's also that they are also getting a bit of radiation from the betas. But we also have then alpha emitters, which are very powerful, but have a very short range. So they get a higher degree of double DNA strand breaks, which is more effective. And that is, of course, something that we're going to investigate in the future, the differences between alphas and betas. But yeah, when it comes to the specific payloads and theories about that, I think that what I know, we're the first ones who are looking into how LRC15 is regulated and what else the LRC15 cells, how they look and what's needed, and the profiling of them. And maybe if we do even more work than we have done, they can figure out a payload for ADC tailored to the specific genomic profile of these cells and tailor a perfect ADC for it.
Mm-hmm.
Yeah.
All right, and then we have one more. Were there any preclinical safety signals to be somewhat concerned about?
As with all antibodies and as with all antibodies that is radiolabeled with radioimmunotherapy or a toxic radionuclide, is that you do have an effect on the bone marrow. But to quote Noah Federman, all of the drugs that are applied today for osteosarcoma already have an effect on the bone marrow. So these doctors that are used to treat osteosarcoma patients, they are very well trained and know how to treat patients where you see an effect on the bone marrow. Mm-hmm. So again, we have tried to make smaller antibodies, sort of fragment antibodies of the DUMP19 antibody, but we don't get the same uptake in the tumors, and the amount of radiation that ends up in the tumors are lower compared to when you utilize a full-sized antibody. So, of course, it would have been better to have a small molecule for imaging, especially because you don't have to wait such a long time to generate the image. So as what I've understood from people who really know small molecules, it is not, from what they are saying, it's not possible to make a small molecule targeting an LRC15. You have to rely on an antibody.
Dr. Ulmert, you kind of mentioned earlier that one advantage of this is that you could kind of select for patients that had high LRC15 expression. So is there a threshold for efficacy? And did any of the imaging correlate to progression-free survival or overall survival in the mice?
That's a very good question. Of course, it's always like more is better when it comes to targeted radioimmunotherapy. You make sure that you get enough radiation into the cell and you are surely sort of causing a DNA strand breaks. But you also have to think about these are very complex diseases. So it also has to do with the rest of the genomic profiling of the cells, how they react to radiation. But it's also pathobiological work that we haven't, we're looking forward to do, but we haven't had the time to do it. in different diseases, you have different genetic backgrounds. And it could be that they are more or less depending on LRC15. So if you're ablating LRC15, even if it's low LRC15, it can have a huge effect in some tumors. We've seen that with, for example, the trial by AstraZeneca, where they go for what they call ultra low HER2 therapy, meaning that they're using a HER2 targeted ADC, and the levels of HER2 are extremely low, but they still see a benefit for the therapy.
And so for the trial eligibility, there is no, you don't have to express a certain amount of LRC15 or anything like that, right? As you just need to have unresectable disease?
From what I understand, and I think that that's a really good question for Lantheus and for Noah Federman. But from what I understand is that you show LRC15 expression in the tumor. But again, I will. That's a good question for Noah Federman, how they're setting up the trial of Lantheus. So reach out to them, and you get a much better response and more accurate response than I can provide with.
Okay, great. And we did share also a link to the clinicaltrials.gov link in the chat too, so people can refer to that for more info. And we're being really greedy with your time, but just a couple more questions. So there's currently a TGF beta inhibitor, a Vactocertib trial for osteosarcoma. And so could that inhibit LRC15? And could that suggest that Vactocertib could also make immune checkpoint inhibitors more effective?
Well, the problems I see it, and the person who probably asked the question is going to try to strangle me. But I think that the problem is that when you're inhibiting TGF-beta, it's very, very, like I said before, TGF-beta in some tumors are helping the immune system to decrease the tumor and inhibit the tumor. And if you then remove TGF-beta activity from that tumor, that specific tumor, it becomes more aggressive. And the other thing is that TGF-beta has so many functions in the body that you are also inhibiting the other, immune systems and repair systems in the body that is depending on TGF-beta. So what I've seen in trials so far is that you're not able to give a dose that's high enough to get a really good response with TGF-beta inhibitors because you are ablating the rest of the TGF-beta in the body as well. But that said, it would be really interesting to pursue when you give drugs targeting LRC15, so targeting inhibiting TGF-beta and combining it with our imaging capacity of this antibody to monitor how it's working, etc. That is something that, so someone wants to give me money to do those studies, we're more than happy to do it.
There you go. Well, we have a virtual hat on Zoom to collect some money for that. That's yeah. So, okay. So basically you're saying it's kind of more specific because it's capturing it at that kind of when it turns into the foe. And then in this case, you're kind of leveraging, you don't want to really get rid of it yet because you want to leverage it to be able to target the tumor cells and the bad stromal cells. Yeah. Okay. One last question. We'll let you go. It's kind of a combo question, but okay. So my understanding on why immune checkpoint inhibitors haven't worked in osteosarcoma is that because they don't usually work when there's a low mutational burden. And so does this debunk that, or is it just like another contributing factor? And I guess moving forward, are you looking at I guess you're doing basically like a dose finding trial right now, but looking at combining it with an immune checkpoint inhibitor?
Yeah, exactly. So we've seen in different models that it's, as I've shown in the presentation, that you do have a really significant effect when you're combining it. Even we used very low doses of LRC15 to see in order to see if we saw any change. But in the case of osteosarcoma, we have not done those studies. So these are the studies that the Kaplan family have, generously sponsored. And we are going to look into the combination with LRC15 with immune checkpoint inhibitors in these tumor models. And again, the caveat of doing academic preclinical research is that mouse models are mouse models, and you don't know how effective it is until you go to the patient. So the patient trials that we're doing right now will be fundamental to determine if you see this change in the tumors in osteosarcoma. And in combination with the trial, or in parallel with the trial that we're doing in osteosarcoma, Lantheus have also set up a basket trial in Australia, where they are also testing uptake in other forms of cancers, such as breast cancer and head and neck, et cetera, et cetera. So we'll see. It looks promising with other models. We'll see if that is as good in osteosarcoma. And if it is positive, you have to think about that it's a mouse model. It's not a human. So you have to be a little bit, humble when you're evaluating the results. So we'll see. Hopefully, that's what I hope for and believe in, that this will be a drug. But we'll see. We don't know until we've tested it in humans, unfortunately.
Yes, but it's great that we're at the point where we can do that. So thank you for all your work, and thank you so much for joining us on OsteoBites today and for making it better for osteosarcoma patients. And especially thank you for being so generous with your time. I think you might hold the record for longest OsteoBites episode. I don't think we've ever gone this long, but it was just so much to discuss. I mean, there's so many great kind of really features, I think, about this therapy that are really interesting and also have implications for kind of other therapies. So again, thank you for all your work on this. More information on this and all OsteoBites can be found on our website at mibagents.org, on our MIB Agents YouTube channel, and at your favorite podcast place. And we hope you can join us next week on February 12th. We are going to be joined by Dr. Caroline Maloney from the Medical College of Wisconsin. She will discuss her research on surgery-accelerated metastasis and developing perioperative therapies. You can find our OsteoBites lineup for the next few months on our website. You can subscribe to our YouTube channel or follow us on Spotify so you never miss an episode. And if you have ideas for future topics that you'd like to hear about, please email us at events@mibagents.org. Thank you again, Dr. Ulmert and Andrew, for spending time with us today. And thanks to all of you for taking time out of your day to join us. We hope to see you back here on OsteoBites next week when we chat with Dr. Maloney. Thanks so much, Dr. Ulmert. Thank you, Andrew. Thanks, everyone.
Thank you. It's been an honor to be on the podcast. Thank you.
Thank you.